which basically means B cells, certain macrophages, or neutrophils. Right? Okay. That's what APCs are. Okay, you still with me? Because we're getting to the fun stuff. Isn't this fun? Aren't you just glad your body just takes care of this for you? And
I don't know what to think about it.
I must say, it would be like a video game. It would be very cool if you could, on your phone, say, okay, I want to activate some T cells. Go to fight, right? Oh my God, that'd be so cool. If you had real-time notifications on your phone, you've just been exposed to an antigen. What do you want to do, you know? Mount B cell, mount T cell, you know, go swimming, or drink, you know? What do you want to do? I think that'd be a great little video game. We might end up all sick at the end of the day, though. Okay, so this is the section where most people lose it. So I want to be sure that you guys understand what Class 1 and Class 2 MHCs are.
What do you have for what is the exception on this one?
Oh, I'm sorry. If they're identical twins. These protein complexes are unique to each of us right now. None of us have the same Class 1 MHC. Okay? None of us. But if I had an identical twin, we would.
Dude,
You do?
I've got twin boys.
Oh, cool. Yeah. So it's nothing but a protein complex. Another part of that glycocalyx, do you remember that word? Another part of that fluid mosaic on the cell surface just means that Class 1 MHC is going to be something that a T cell wants to recognize. So let me say this before I get too involved. If you guys come out of this lecture today, and even after you listen to it, and you're still clueless, like, what did he do? What did he talk about? At least know this, right? Because this is the meat of it all. Can you link these words together? Cytotoxic T cells have CD8 receptors, which are sensitive to Class 1 MHCs, which are found on all body cells. 1, 2, 3, 4. Link that together, right? Dissect that when you get a question, because that's really what it comes down to. There's no video on the test. I'm not going to ask you a series of questions that are so detailed that you don't have a shot. This is what you need to know right here. That's why I did that first. Yeah. Say it again. Sorry.
The Class 1 MHC protein, does it tell if it's a self cell?
Yes.
Does the T cell recognize
That's a great question. The Class 1 MHC means that is a normal, regular, healthy cell. All cells have it. Right? So when T cells look at a cell, they recognize the Class 1 MHC. They see that it's there, that means it's not going to mount an attack. Right? Great question.
It's like,
Unless that Class 1 MHC is bound to some kind of different protein where it looks different, then it will mount an attack. So if the Class 1 MHC has along with it another foreign bound protein, an antigen fragment, right, then it will mount an attack. Right.
so it just checks all those, and if it is something
If it
wrong-
notices something different from the normal MHC, then it will mount an attack. Right. In the case of autoimmune diseases, it looks at the Class 1 MHC and says, that's foreign, even though it's not. So then you got immune cells killing all of our cells, which is not good. That's the disease state, right? They don't want that. But in a normal circumstance, it's recognizing the normal self-antigens. These are normal, by the way, if I missed saying that. Class 1 and Class 2 MHCs are normal protein complexes on cell surfaces. We just have to know where they are. Class 1's on all cells, Class 2 is on B cells and APCs. So if there's anything different about the Class 1 or Class 2, something is altered, something else is bound to it, then it knows to mount an attack. Okay?
If the B cells are doing the plasma cells and the antibodies, then it'll get the T helper cell to come and kill it.
Yeah. Once the B cell binds to an antigen, we'll see it in the video.
It's different.
Once you see it, it'll make a lot more sense. Once this bounds to an antigen, right? You'll see how their helper cell comes up and starts doing its stuff. Another question? Just wait for the video. That might help. There's only so many words I can say. I can't paint the whole picture until you finally see it. Okay, so cells of the adaptive immune system, we know we have B, we have T, and now the APCs, which by themselves used to be part of the second line, right? That's funny because in New Orleans, the second line is something different. You know what that is? Okay. That's when they all dance in a party. They just walk around the room. Y'all never heard of the second line? Wow. The big Mardi Gras thing. You take a napkin, right? And you drink. And somehow you still hold on to the person in front of you. And you dance that song. Dun, 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 dun. You just go in a circle acting stupid with your umbrella somehow. So you got your drink, your umbrella. Anyway, that's a little addition. So APCs act in two places. They can act on their own, and then they can act in concert with the T helper. Okay? So that's why it says, do not respond to specific antigens. They just eat everything. Right? They don't have to worry about binding to a specific antigen. It just notices that it's foreign and it eats it. As it eats it, it digests it inside the lysosome and then puts out a little fragment on its cell surface and presents that fragment to a T helper. That's why it's called an antigen presenting cell, because it's presenting a piece of the antigen to the T helper.
Okay, that was my question. That's what threw me off the class 1 MHCs, because I thought only the class 2 were the ones who put up the flags. So would that be like when your body attacks itself with the class 1?
No, you'll see it. I know what you're saying. The difference is that here, the B cell is presenting the fragment, or the APC is presenting the fragment to the T helper. On the cytotoxic side, each tissue cell itself can bring in an antigen, or can be infected, I should say, and present a fragment. So it is its own antigen-presenting cell, if you want to think of it that way.
Okay.
Right? So a virus comes into a normal tissue cell, let's say in the lung. That tissue cell absorbs that virus, right? The virus starts making proteins on the inside of the cell. Those proteins are foreign, so those proteins are sent to the cell surface to say, hey, I've been infected. So now that class 1 MHC on a regular old tissue cell will look a little bit different now that it's infected. So then a cytotoxic T cell will come around, notice the class 1 MHC is altered, and then kill it straight out.
The entire cell?
Yep.
Okay.
Yes. In the back.
Okay, did you tell us what triggers the APC to eat it and show them?
APCs they're just macrophages.
So they do it on their own?
Yep. They're non-specific in that case. They just eat whatever's foreign. It doesn't need to be a specific shape like we saw the antibody needed to match to. Yeah, these are great questions. You guys like the first class I've had so many questions. It's wonderful. Okay. Now, we mentioned these two words for immunocompetence and self-tolerance. This is where we learn how to recognize self and how to not attack the self, right? So we don't want cytotoxic T cells recognizing class 1 MHCs and then attacking them, right? That would be horrible. So two things. It seems obvious, but it is two different learnings. It has to recognize the class 1 MHC and then also ignore it, right? Not mount an attack. So that is important. If, in case it does start to mount an attack on a normal cell, other white blood cells around it, other T cells around it, will notice, hey, he's not following the rules. Let's get him. And that's what happens. Another cell will come by and put a little protein on its cell surface, tagging it for death. And then a macrophage will come and swallow it. Right? So yeah, that's happening like right now. So pat yourself on the back for being that cool. It's very cool stuff. There's so much about the immune system that's very much like actual battle strategy. Okay. So we know what these are. We know what that is. APCs, now we know all about APCs, right? Let's do the data. APCs can be dendritic cells, which are just macrophages that are all around the body. Activated B cells, we learned that. What do they have? Class 2 antigens, don't they? Class 2 MHCs. Check. Everybody got it?
APCs have class 2 MHCs.
Beautiful. Yes, they do. APCs have class 2 MHC protein complexes on their cell surface. That's how the video says it. And what do they respond to? CD4 receptors. See, we can twist these words like 1,000 ways. That's why right here, this is you. This is where you're going to swim and know how to put these words together. Written on the board. Okay. When an APC presents the antigen to a cell, what does it do? To the T cell? It activates the T cell. When that T cell has been presented the antigen, it starts activating different things inside the T cell, and then they're going to secrete something called cytokines, which are going to be other chemicals that can enable other T cells around them to start acting different. So it's like a priming. Does that make sense? But that only happens when it's getting bound and presented. Right. Right. So in a sense, the T cells are going to mount a bigger response once they're bound. So here's our terms. Our third line of defense gives us either humoral, which is antibody-mediated immunity, and that's for what? B cells, or we call cell-mediated immunity, and that's what the T cells do. It's just two different pathways, you could say, for immunity. So isn't this valuable stuff right there?
Yeah.
We keep pointing back to it, so I would kind of know that on the board there. This can get you out of the weeds. If you don't understand anything on what I'm talking about today, at least know the screen, the board. That can actually mean a lot. Okay, so the antigen binding will trigger what's called a clonal selection, which means that antigen will bind to a certain antibody, right? Or an immunoglobulin receptor on a B cell, same kind of thing, which will trigger it to do what? Clone, right? The B cell will grow rapidly. Most clones become plasma cells that will secrete antibodies, right? And few clones will become memory cells. Perfect. Now it's getting clearer. So here we go. Pay attention to this picture. There's a lot going on, but if you take it step by step, look at what's happening at the very top of the screen here. That little red dot represents an antigen. Is it binding to each one of those B cells? No. And by the way, notice the Y shape on the B cell? That is an immunoglobulin bound to the B cell, right? That's in a sense an antibody. Okay, immunoglobulin is an antibody. Bound as a receptor on the B cell. Okay? That receptor, notice only the B cell in the middle has the right shape antibody on it to bind to the antigen. Notice the antigens don't bind to the one on the left or right. Right? They're ignored because it's not the right shape. So now once it's bound, what happens? It says success, right? We have a correct binding, so it starts to clone itself because it succeeded in binding to an antigen. So it wants to clone that. All those clones, some will become plasma cells, right? These plasma cells will secrete those immunoglobulin receptors that were up here. Now it lets them loose in the blood. That's why we call it humoral immunity. So plasma cells lose their receptors that they originally had up there, right? Secrete these antibodies, and then some of that population of clone will become memory. Okay, perfect. So after this, we'll look at the video. On a secondary response, could be years later, months later, now that same shape antigen, right, comes around, binds to the memory cell receptor, immunoglobulin receptor right there, and what happens? Now the cloning is even faster. Look how many clones we get versus the first exposure. right? Because it's already primed up.